Okay, guys, we're ready for part 2 of our macromolecules lecture. And so now I'm going to talk about nucleic acids and proteins. And so nucleic acids is usually something that everyone is really familiar with, something they truly remember from introductory biology. And so we know that nucleic acids are our genetic material, okay? They're going to store all of our information. They're the blueprint of our body. They are going to tell exactly how all of the proteins that function for life are going to be built. And we know that we do that. DNA goes to RNA, goes to proteins. That's our central dogma. And so we transfer information. And this is what we pass on to the next generation. So your DNA is what you're going to pass from parent to child. And so nucleic acids, we have two different types. We have RNA and DNA. RNA is ribonucleic acid, and it's made of just one helix, and then DNA is deoxyribonucleic acid, and it has a double helix. And so the polymer is going to be nucleic acid, but the monomer is a nucleotide. And so let's talk a little bit about nucleotides. They're made of 3 parts. And so these 3 parts are composed of carbon, hydrogen, oxygen, nitrogen, and phosphorus. So this is kind of like our greatest number of different elements or different atoms that are going into a single macromolecule. And so the first part is the nitrogenous base. And a nitrogenous base is just going to be a ring of carbons and nitrogens. So we'll have like a carbon ring, right, with some nitrogens off to the side of it. Then we will have either a pentose or a ribose sugar. We will have a pentose sugar. It's either a ribose or a deoxyribose sugar. And so pentose sugar just means it has 5 carbons on it. And then we're going to have this phosphate group on it as well. And so the nitrogenous base is what you think of when you think of like DNA code. That's going to be A, T, C, G, or U. And then the phosphate group, if you notice, okay, it has this charge on it. Okay, so we saw this phosphate group, right, when we looked at the phospholipids. And so we know that phospholipids, they have that affinity for water because they are negatively charged. And so that's going to be the same thing with DNA. So DNA is a charged molecule, and it is negatively charged. There's 2 types of nucleotides. So we have purines and pyrimidines. Purines are going to have a double nitrogen carbon ring, and then pyrimidines are going to have a single carbon nitrogen ring. And so A's and G's are purines, and C, T's, and U's are pyrimidines. We will talk about this a lot more when we talk about DNA replication and things like that. But it's good to just kind of get an introduction to it. If you're looking for a way to kind of memorize or remember which are purines and which are pyrimidines, you could think purine is equal to Ag, or like pure silver. And so pairing of nucleotides, so nucleotides, they bond between, or the nucleotides bond between the 2 strands of your DNA double helix. So we know that A's and T's go together, G's and C's go together. And so G's and C's we should have a couple extra dots there. They have 3 hydrogen bonds, and A's and T's have 2 hydrogen bonds. We talked in our last lecture about how it's important that we do have these hydrogen bonds that link the bases in DNA, because we can easily break hydrogen bonds for things like DNA replication and transcription. And so when we talk about how they all kind of fit together, you're going to have the nitrogenous base that's connected to the sugar, that's connected to the phosphate. And then this phosphate is going to connect to the next sugar, which connects to the next phosphate. And so each of these is a single nucleotide. And so the polymer grows in one direction from 5' to 3'. And again, we'll talk about that more when we talk about DNA replication. But what we're talking about is a 5', the 5 carbon bonds to the 3 carbon. And so that's why we call it 5' to 3'. And this link between the phosphate and the sugar is called a phosphodiester bond. And we call this usually like a sugar phosphate backbone. The sugars and phosphates that are all connected together that make up kind of the rung of, or, you know, the side of the ladder or the rung of the ladder. And so a DNA molecule, it's a double helix. And so it forms that helical structure because of the way that the phosphates and sugars connect to each other, and because of that 5' to 3' configuration, one strand is going this way, one strand is going that way, and so that's why it forms that helical structure. And so here's just kind of like a simple overview of DNA to protein. Again, we're going to get more into this second semester, but you know, we have our DNA, which is transcribed to mRNA in the nucleus. The mRNA exits the nucleus and finds a ribosome, and then is synthesized into a protein through transcription. And you know, we are going to talk about this in much more detail next semester. Okay, so now we're going to move on to proteins, which could arguably be the most important macromolecule, because proteins really do just about everything. And so proteins are very diverse. They make up a ton of different things in our cells, in our body system. So they're going to make up enzymes. They're going to provide structure for our cells. They're going to carry molecules and transport molecules through our bloodstream and in and out of our cells. They are they communicate from cell to cell, so hormones and things of that nature. They also are cell receptors. They help with our immune system and antibodies. They help our muscles move with actin and myosin. And then they're also going to do some storage for us. And so proteins, if you aren't sure what's responsible for something in a cell or, you know, the function that our body does, you can pretty much bet that it's going to be a protein. And so proteins, the monomers are called amino acids, and they're called amino acids because of this amino group that they have. Amino, when you think amino, you can think nitrogen. And so that's kind of how they get, and then the acid comes from this carboxylic acid that's on the other end of each amino acid, and so that's where they get their name. A polymer is a polypeptide, and so that's when there's one or more amino acids that form together to make a protein. They're very complex molecules. We're going to have 4 steps to kind of build them or to shape them and to what they're going to become, and we'll talk about all of that. So amino acids have 3 parts to them. They have a central carbon, okay? So the central carbon that's right here, which is going to be connected to an amino group. And remember, amino has that nitrogen. It's going to be connected to a carboxylic group. So that's C double bonded to an OOH. And then it's going to have this thing called an R group, okay, or a side chain. And that's going to be connected to that central carbon. And so this group is what changes, and this group is what gives us all the different amino acids that we have. We have 20 different amino acids. And so each amino acid is going to be a little bit different based on its R group. So here are the different types of amino acids. And so one group of amino acids that we're going to have are nonpolar, meaning that their R group or their side chain will be nonpolar. And if we look at these side chains and we're trying to, pick out why would they be nonpolar, we see kind of what they're made up of, okay? Single hydrogens, carbons and hydrogens connected together, carbon rings. Here we have a sulfur in there, but again, the sulfur is part of this longer carbon chain. So we're thinking of things that are usually very nonpolar, having nonpolar covalent bonds. And so that's why these amino acids are going to be deemed nonpolar. And so polar amino acids, okay, are going to have charged groups, and they're going to be water-loving, remember. And so when we look at polar amino acids, and we're looking at what kind of groups they have on them, we're seeing these OHs on the end, which we know are usually polar, okay? Or these sulfur groups on the end not connected to any other carbons, or these really electronegative oxygens hanging off on the end, okay? And so that's what is going to make those groups polar. Now, some amino acids are not only going to be polar, but also electrically charged. And so what that means is that they're protonated so that they can either combine to become acidic, or they could combine with something else to become basic. And so again, when you look at these R groups here, we have carboxylic acids on the end. And we know that carboxylic acids normally form acidic solutions. And then if we look here, we have a bunch of nitrogens on the end, okay? And we're thinking amino, like ammonia. We know that that's usually leads to something basic. Now, you are not going to have to memorize all of these amino acids. If you become a biochemist or take biochemistry one day, you will have to memorize all the amino acids and their differences, but not for AP biology. And so just as long as you know that, polar amino acids like water, non-polar amino acids hate water, as long as you can remember those types of things, you'll be completely fine in terms of AP biology. And so something that proteins have that are different from all the other macromolecules is sulfur atoms. And so these amino acids with sulfur in their R groups, they're going to form these things called disulfide bridges. And so pretty much when we'll talk about how they exactly interact with when we're kind of folding proteins into their structure. But these disulfide bridges, they work as a stabilization for proteins. And so all that means is it's just when two sulfurs make a bond together. And so what's kind of interesting about disulfide bridges is that those are actually those are the bonds that you're making and breaking when you curl or straighten your hair. And so they're really susceptible to heat. And so when you curl your hair, and your hair is just a big protein, when you curl your hair and you get those disulfide bridges hot, you break them. You break them like momentarily and then reform them back into whatever structure you want. And so that's how, your hair holds a curl or holds a straighten with that heat and the breaking of those, the breaking and remaking of those disulfide bonds. And so when we build proteins, we're going to do the exact same thing, a dehydration synthesis reaction. So here we have our H and our OH group. The hydrogen comes from the amino group, and the OH comes from the carboxylic acid of the amino acid next to it. We lose water, and then we form something called a peptide bond. And so when we build proteins, they have direction. They go from N-terminus, the N-terminus, to the C terminus. And so N terminus just means that amino end, and the C terminus means the carboxylic end. And so what happens is amino acids can only be added in one direction. And so they can only be added to that carboxylic end. So when you're building a protein, that's where they're going to add on to. Okay, so let's talk about protein structure and function. And so the structure of a protein is going to determine the function that it serves. And so if it's twisted or foiled or coiled, all those different proteins, because of the shape they are going to have different jobs in cellular processes. So how do we get to this structure? The primary structure of the protein is just the order of amino acids. So, I have a histidine and then a lysine and then a glutamic acid. The order that those amino acids go in is just that primary structure. And so, sorry guys. And so the smallest change in an amino acid can make a huge difference. And when I'm talking about changes, I'm talking about mutations. So those mutations can happen in the DNA code that's coding for the proteins. They can happen with a mistake in transcription or translation. But all it takes is one change to significantly change a protein. And so in sickle cell anemia, which is a genetic disease, normally your red blood cells are a nice little donut shape like that, and that's because of a protein called hemoglobin. Okay, so hemoglobin, it sits very nicely on this red blood cell, and what it does is it's a little receptor for oxygen. And so in people with sickle cell anemia, they have a change in one single amino acid. And what that one single amino acid does is it makes it so that hemoglobin no longer has that nice round structure to hold oxygen in, and it makes these red blood cells sickle, or kind of, make that crescent moon shape. And so what that does is that leads to a lower oxygen concentration that those red blood cells are carrying. And it can also cause buildups in capillaries, because all these blood cells that normally are circle and would move through a really tiny capillary get stuck because of that crescent moon shape that they're in. And all of that is just because one single amino acid was changed for the hemoglobin protein. So the secondary structure, that we'll refer to that kind of as local folding. So pretty much it's just when the amino acid chains start getting connected with amino acids that are close to it through hydrogen bonds. And so there can be two structures that they form, an alpha helix or a beta pleated sheet. And so an alpha helix is going to look something like that. And so this is just amino acids that are close to each other. There are groups forming hydrogen bonds with other R groups. And then over here we have a beta pleated sheet. And so if you look at this protein that's right here, you can kind of see like you'll have an alpha helix and then maybe a part of a beta pleated sheet connected to another alpha helix and then another beta pleated sheet. And so it's just kind of random, and it's just wherever these hydrogen bonds are forming. The tertiary structure, or the third step, would be that whole molecule folding. And so this is where we're going to see more interactions with R groups, but R groups that are farther away in the amino acid sequence from each other. This is where we're going to see that hydrophobic and hydrophilic interactions. And so here, if you have this protein, if this is like the entire protein right here, the amino acids that end up forming on this outer edge are going to be things that love water, okay? They're going to be hydrophilic because these proteins, right, are going to be in the cellular space where you have an aqueous solution, and so they're going to have to be water-loving. And then the water-fearing amino acids are going to try to turn themselves in so they have less interaction with that outside aqueous space. And so how do we form this tertiary structure? It's just through hydrogen bonding, through ionic bonding, and then through those disulfide bridges. So here you have two R groups connecting with a hydrogen bond, you know. Here you'll have an ionic bond, and then here you'll have kind of that disulfide linkage. And this is really going to be what really holds that shape. These are going to be, you know, like anchoring that 3D shape that the protein is going to have. And then the quaternary structure of a protein is just when you take more than one polypeptide chain and put them together. And so in the case of hemoglobin, you're going to have 1, 2, 3, 4 different polypeptides that come together to make this protein that has this large circle shape. If we're talking about something like collagen, we're having, you know, kind of these 3 different protein chains that are all interwoven together to give collagen its quaternary structure. And so again, like these are going to be those hydrophobic interactions that orient these protein chains so that, you know, their hydrophobic amino acids are facing outward. And so the last piece is this idea of protein denaturation. So what happens when you have a folded protein, it's all together in its quaternary structure, like what would disrupt that? And so what's going to disrupt that is anything that can mess with those hydrogen bonds, ionic bonds, or those disulfide bridges. And so just like when I was talking with the curling iron, temperature is something that can, you know, break and then remake these bonds. pH is huge. So if a protein enters an environment with too high or too low of a pH, you know, it will make those bonds, those, you know, tertiary bonds break and start to unfold that protein. And then proteases are enzymes, and what they do is they go and they're just like scissors and they just cut proteins up. And so again, like that's going to take a folded protein and unravel it. And so usually this is altering like those beta pleated sheets or that tertiary structure. And so what it's going to ultimately do is it's going to destroy the function. And so after a protein is denatured, many cannot refold themselves to come back. And so, you know, whatever that protein was doing, it's no longer going to be able to fulfill its job anymore because it's unfolded. And so that's why, you know, pH and temperature and things like that are so important when it comes to bodily functions and our cells and them being able, you know, to survive. And so that is kind of the last thing we have to talk about for proteins. Again, if you have any questions, you're going to want to bring those to next class when we have a class discussion about macromolecules.